All right. Hello and welcome, everyone. Thank you for joining us this morning or this afternoon or evening. My name is Andy Bredemeyer. I'm excited to welcome you to Velsera's webinar, Interpreting the Tumor Genome, Leveraging a Medical Knowledge Base Built for Purpose. Velsera connects healthcare and life sciences to reveal the true promise of precision medicine, a continuous flow of knowledge between researchers, scientists, clinicians around the world, creating insights that radically improve human health. I will briefly introduce our speakers today and then hand the show over to them. Please, during the webinar, at any time, you may send questions to me in the chat. You can select my name there from the chat window and put any questions in, and we'll leave some time at the end and answer the questions that you've put there. I'm pleased to introduce my Velsera colleagues today. First, let's say hi to Dr. C.D. Rahul. C.D. is a senior biocurator on the research and development team at Velsera within the Knowledgebase group. C.D. designs, develops, and maintains strategies to represent clinically relevant genetic data, ensuring high quality and data accuracy of Velsera's knowledge base. C.D. won the gold medal for outstanding performance during her Master of Technology in Biotechnology, and she trained as a research fellow at the Tata Institute of Fundamental Research in the Department of Chemical Science and earned her PhD in cancer biology from ISER, the Indian Institute of Science, Education and Research in Pune, India. Please also welcome Aditya Muley. Aditya, who is a senior biocurator on Velsera's knowledge base team, specializes in comprehending and reviewing diagnostic, prognostic, and therapeutic information related to cancer. This involves analyzing various sources like clinical practice guidelines, drug labels. Aditya plays a critical role in developing curation strategies to create evidence-based interpretations for biomarkers associated with different types of cancer and drug management using information from clinical practice guidelines and drug labels. Aditya pursued a master's degree in biotechnology and gained further expertise through research fellowships at the University of Cologne, Germany, and at the Tata Institute of Fundamental Research, where he focused on developmental biology. C.D., Aditya, thank you for your time today. We look forward to your presentation. And C.D., I'll hand it over to you.
Hey, thanks, Andy, for the introduction. Hello, everyone. Welcome to the webinar. In the next 1 hour, Aditya and I will give you an overview of the Velsera's knowledge base and how it drives interpretation of clinically relevant genomic variants. The three focus areas of today's session are: first, how Velsera's knowledge base empowers you by providing access to a wide range of clinical data that aids in your decision-making processes. Second, how our intelligent and adaptive learning algorithm ensures you deliver the best patient outcomes. And third, Velsera provides you with a shared interpretation knowledge base where you can leverage the knowledge of clinical community for reliable and accurate clinical reporting and discovery. In short, Velsera's knowledge base provides the healthcare practitioners with a comprehensive clinical data access, enabling them to make informed decisions, increasing the accuracy of the clinical reporting and ultimately leading to the best patient management decisions. Now, the inception of Velsera knowledge base has its root in the fact that our genetic makeup can influence our susceptibility to certain diseases. Not just the susceptibility, but it also governs how a specific individual will react to treatment options or a therapy. Velsera knowledge base integrates this genomic information into healthcare system, enabling or facilitating more precise and effective intervention, leading to improved patient management decisions. In line with the previous rationale, another need for a Velsera knowledge base or for such a clinical grade knowledge base is that new breakthroughs in clinical genomic and precision medicine are happening every day. With increasing use of large NGS panels to detect multitude of genomic alterations in tumor samples, it is imperative that laboratories are equipped with quick identification, reviewing, and prioritizing of variants which are clinically relevant. Velsera leverages human curation along with algorithms and intelligent algorithms to sift through this large amount of data which is available, and it brings together the most pertinent and up-to-date clinical information from diverse sources. It converts them into an easily accessible format or data in the form of a clinical knowledge base. Velsera's knowledge base is also unique in the fact that it is built up on an adaptive and intelligent learning algorithm model. When I say that, what do I mean? I mean that in other variant lookup tables or knowledge bases, when you are looking for information about a variant, you get information about, okay, this variant has effect in this particular disease. It may respond to this particular therapy option, or some clinical information related to that specific variant or that specific um, disease condition. Now, Velsera's knowledge base not just has information about a particular variant and disease condition, but it also stores a lot of additional information surrounding that variant. Say, for example, what domain that variant lies in, what exon that variant lies in. Is the variant, or that genomic event always occurring in a standalone fashion in a disease? Does it occur with another genomic variant? Is there a co-occurring scenario? Is it a secondary mutation or is it a primary mutation in a disease scenario? So all this metadata is also available along with that variant when you are trying to look for information for the variant. So this kind of makes Velsera's knowledge base intelligent and adaptive. That's what we mean when we say we follow this intelligent adaptive algorithm. Now, when I spoke in earlier slides that there is a lot of data available and Velsera's knowledge base, curates information or covers information from all these diverse knowledge base or diverse sources of information, what exactly am I talking about? So Velsera is based on three main arms. The first arm of Velsera knowledge base is the expertly curated content. Now, expertly curated content means that we have our curators or human curators, which are trained under quality controlled SOPs. And these expert curators go through the clinical practice guidelines along with approved labels and collate information about the available therapy options or the available prognostic information related to a specific variant or a biomarker, and store it out or populate the knowledge base. Now, apart from clinical practice guidelines or approved labels, our curators, expert curators, also go through or collect information about investigational and emerging therapy data from published literature and populate our database. Along with that, we also have information about the recruiting or the available clinical trials that are, recruiting a specific patient with a specific variant or a specific biomarker. So all this information, which is curated by our expertly trained and highly trained curators, is present in our knowledge base. Now, if you are looking for information about a specific variant, then you also are provided with a ready-to-use interpretation, which has available information from all these different sources. Then you can build upon the interpretation, or you can use it to, write your own interpretation about variant of interest. The second arm of our knowledge base is the real-world medical content. Now, when I say real-world medical content, I mean that we have our in-house interpretation service, which writes interpretations for a specific variant which was detected in a real patient in a disease scenario. This information, upon sign out of the clinical report, is de-identified and stored back into our knowledge base. Apart from our in-house interpretation service, we also have, like I mentioned earlier, a shared interpretation knowledge base, which means that we have all our clients can, have right interpretations about their variant of interest. And when they sign out the clinical reports, these interpretations about the variant are de-identified again and stored back into our knowledge base. So tomorrow, if you are looking for information about a variant, which is kind of a novel variant, you will get access to the data which has been previously identified or written down for that particular novel variant and stored back in knowledge base. And then you can build upon that stored information from real patient data and write your clinical reports. This kind of makes your interpretation of the variant that much reliable and accurate. The third arm of our knowledge base are the publicly available data sources. We have information about a specific variant or a biomarker populated from public databases such as ClinVar, Cosmic, and OMIM. Additionally, we are also interested in giving out information that what is the frequency of this particular variant, like how frequently is this variant identified or detected in real world scenarios. So we have incorporated databases such as Nomad and Exact, which gives out information of minor allele frequencies, which also help you identify whether your variant of interest is polymorphic or not. So together, all these three arms, form or populate our clinical grade genomic knowledge base. Now we have a lot of, I spoke about we have diverse sources of information which populate our knowledge base. I also spoke about how we have an intelligent algorithm which stores information about the variant, not just a specific variant, but also all the metadata. Now, how does it help you, or how does that gets translated into patient management decisions? So for that, knowledge base follows the if-then rule condition. So if a specific set of rules are met, in our case, whether it's a specific gene or a specific variant or a specific fusion partner identified in a specific disease condition, then specific set of clinical assertions, which the umbrella term clinical assertions, when I say, I'm talking about the therapy options, the prognostic information, the information about any recruiting clinical trial or any emerging therapy options that are available. All this clinical assertion is populated if this set of conditions are met. That is how our information of knowledge base is converted, or rather available to use in a clinical reporting system. We have almost 350,000 such rules in place in our existing or current knowledge base. Now I've added an example here just to explain how our if-then condition works. If you have a patient that has EGFR insertion variant and the disease of the patient is lung cancer. So the if condition becomes the disease is lung cancer and an exon 20 insertion rule. If these conditions are present in a knowledge base, then the information that is populated or fetched from the knowledge base is that, okay, if your patient has this particular variant and this particular disease, then they may benefit from this therapy option. Along with the clinical assertions, the source of that clinical assertion is also populated for you. Say, for example, whether this information is coming from a clinical practice guideline, or whether it's coming from an approved label, or whether it is an emerging therapy option which is coming from a published literature. So all this source also helps you build confidence on the accuracy and reliability of the information that you are signing out for that particular variant. Now, in order to exemplify better, when I say that we have a flexible and intelligent model, what do I mean here? What I want to say here is that we not just cover or give information about a variant or a syntax, but we also have information and knowledge base at a codon, exon, or an whole gene level. So, for example, if you consider an example here of EGFR, we have information about the entire exon 20 insertion event. So it is it's widely known in clinical community that in-frame exon 20 insertion events are resistant to the first generation inhibitors, EGFR inhibitors. So this information is present in our knowledge base. So this kind of information helps making knowledge base not just present perfect, but also future ready. When I say this kind of domain related information makes our knowledge base future ready, I mean that, say for example, tomorrow a novel variant is detected in your patient, which is exon 20 insertion, like it falls in exon 20 and it's an in-frame insertion event. However, if you go to look for information about that specific variant, you will not find much clinical relevant information for that variant. But because it's already known in the community that in-frame insertions in exon 20 might be, resistant to the first generation TKIs, the knowledge base is smart enough to populate this information for your novel variant. So when you try to find information for this novel variant, the knowledge base will populate that your patient, because it's an insertion in exon 20, might not benefit, or might be resistant to these therapy options. Along with that, the information will also be populated as to what are the other therapy options that might be available for your variant or patient. Aditya will be talking more about this scenario when he discusses case study later on. Another feature of a knowledge base, which kind of makes it unique in the sense of, when I talk about intelligent and adaptive learning algorithm, is that we also have the option of storing information about co-occurring genomic events. When I say co-occurring genomic events, it's well known that many a times one or more genomic, like two or more genomic events can occur at the same time in a single patient. And the management, or line of management, or the management decisions change or vary based on whether there is a single genomic event or two genomic events are co-occurring. This kind of information is also stored in our knowledge base and can be leveraged by the users when they are trying to report or generate the report for their variant of interest. If you look at the example that I've cited here for the co-occurring scenario, your knowledge base has this information that if ATRX gene loss and IDH2 mutations co-occur, then it is diagnostic for astrocytoma. So if a patient walks in who has both of these mutations co-occurring, then the knowledge base will give you an information that this looks like a diagnostic scenario for astrocytoma. Such kind of co-occurring scenario information, along with all the other metadata model that I spoke about, makes our Velsera knowledge base kind of future ready and present perfect. Okay, now I spoke about what are the diverse sources that are present in our knowledge base. How does information get populated for your variant of interest from the existing knowledge base information? The next thing is, how does this information help you identify actionable variants in your patient's disease? So knowledge base follows this AMP ASCO CAP classification algorithm, in which we classify variants based on their tier levels using the expertly curated content or interpretations from real world data. And for tier 3 and 4, that is polymorphic, or for the variants for which information is not available as much, we use the available public databases to classify those variants. So tier 1 and 2 variants, which are variants of strong clinical significance or potential significance, are classified based on the content which comes from clinical practice guidelines, approved labels, or emerging therapy. And it also comes from the real world interpretations that are present in our knowledge base from previous encounters of the same variant, in the real world patient data. And the tier 3 and 4 are classified, as I mentioned earlier, based on our population databases or the publicly available databases that also populate our knowledge base. So this kind of gives you a confidence that the variant you are looking for, if it is a tier 1 variant, then it gives you a confidence that, okay, the clinical assertion that I'm reporting out for my variant of interest is reliable and accurate, as it comes from a tier 1, which is expertly curated content or real world scenario. In addition to identifying actionable variants, somatic variants in a patient data, Velsera knowledge base also has this algorithm incorporated, which helps biological classification of your variants. So this is a ComperMed guideline algorithm, which is present in our knowledge base, wherein there is a consensus pathogenic variant list, which is available or published by the ComperMed guideline. And if your variant falls in this list, then it automatically is classified as a pathogenic variant. However, there are few exception rules to this ComperMed algorithm classification. And these biomarkers of variants which fall in this exception rule are classified using information from our knowledge base. Now, both these classification systems, that is the tiering as well as tiering for actionability of the variants, as well as the biological classification to understand the variant better. Both of these are editable, or both of these are flexible in the sense that users can change the classification system based on either their understanding or their previous encounters of the same variant. And this changed classification system is also then stored back into our knowledge base and is available for use in the future if you are interested or working on a similar variant. Now, I have spoken about, um, in theory, what populates a knowledge base and how it helps you in identifying actionable variants, and it helps you be future ready. However, this is patient care that we are talking about. It's a patient that we are talking about, and we do not take the quality of the content that we are populating in our knowledge base lightly. We ensure that the quality of our knowledge base is at par with the international standards which are recommended for patient management systems. And Aditya will be taking over and discussing with you all the quality control and quality assurance systems that we have kept in place for establishing, or for ensuring that the quality of content that we are providing in knowledge base is uh, it's at par with the standard that is required. Additionally, whatever I have spoken in theory till now, Aditya will also try to explain the usability of all those things by using a case study, a synthetic case study that he has designed, so that you get a clearer picture of how the knowledge base will help you in interpreting your variant or genomic event of interest. Over to you, Aditya.
Hey, thanks, Siddhi. Yeah. So, till now, we have discussed the valuable role played by knowledge base in identifying actionable variants. However, it is crucial to emphasize the importance of implementing stringent quality checks to ensure precise results. It is therefore imperative that we use rigorous quality processes to achieve. And to achieve this, we diligently adhere to ISO standards, meticulously follow documented processes, and conduct thorough content accuracy validations. Next. Siddhi, next slide, please.
Hey, hold on a second, Aditya.
Yeah, we are back. Okay. So along with these quality management systems which are used, Velsera also implements stringent quality workflows for content curation. These workflows ensure that the highest standards of accuracy and reliability are maintained. So there are some key elements for these workflows, which are: first is to have a training module and proficiency assessment for our curators. What I mean by that is, to maintain the consistency and expertise among our curators, we have developed standardized training modules. These modules cover essential knowledge and skills which are required for effective content curation. Additionally, regular proficiency assessments are conducted to evaluate the curator's performance and ensure ongoing competency. Second is, these trainings are governed by the ISO 13485 compliant processes, which ensures that they align with internationally recognized standards for medical device quality management systems. By adhering to these processes, we ensure the highest level of quality in training and development of our curators. Third is having a standardized SOP. So to establish, again, to have a consistent practice and workflow, we have implemented standard operating procedures. These SOPs serve as guidelines for the content curation process, and they undergo rigorous validation to ensure their effectiveness. Moreover, these SOPs are version controlled, ensuring that the latest validated versions are followed consistently. And the last key here is to have an expert review of the curated content. So our team of subject matter experts conduct thorough reviews of the curated information. Their expertise and insights help verify the content's clinical relevance, accuracy, and adherence to best clinical practices. Next. Furthermore, to ensure the accuracy and comprehensiveness of our content, we have implemented the following quality assurance measures. First is to measure the accuracy or to maintain the accuracy of assignment of clinical relevance. We evaluate the performance of our knowledge base by comparing the auto classification of variants within the knowledge base to the variant classifications which are assigned by our medical directors based on finalized reports. This assessment is conducted based on relevant tiers like tier 1A, tier 1B, tier 2C, as well as tiers 3 and tier 4 variants. Then second is to check the sensitivity and accuracy of reporting clinical associations. To enhance the quality of our knowledge base, we employ orthogonal database testing. This involves utilizing independent databases such as Civic that are considered to represent the ground truth in terms of clinical relevance, which includes therapeutic, prognostic, and diagnostic claims as well as classifications. While this assumption has its limitations, it enables us to calculate objective metrics such as true positive concordance of the knowledge base. Tracking the overall performance of the knowledge base using these metrics helps to identify any issues that may require adjustments or improvements. And the last key here is to have a knowledge base expert panel. Our knowledge base expert panel, it comprises of external experts like medical directors and pathologists, and they play a vital role in ensuring the accuracy and reliability of the knowledge base. They work collaboratively to establish and review consensus on strategies for handling ambiguous high importance biomarkers within the knowledge base. This process is conducted in alignment with the best clinical practices, providing valuable insights and expertise to enhance the content's quality. Next. Now let's examine some case studies that highlight the practical utility that we have been talking so far. So in this hypothetical case, we have a patient diagnosed with non-small cell lung cancer, which is a subtype of lung cancer. And it is that patient is presented with various biomarkers, which includes both well-known mutations associated with NSCLC and some novel mutations. By utilizing the knowledge base, we can derive valuable clinical information indicating that this patient is expected to respond favorably to BRAF or MEK inhibitors and RET inhibitors. This assessment is based on the presence of BRAF-P V600E mutation and a RET fusion. Next. This section provides a holistic overview of the overall clinical findings, with auto-classification assigned to variants based on the available clinical information in the knowledge base, specifically for NSCLC or any other cancer types. It demonstrates how the knowledge base categorizes and classifies these variants according to their clinical outcomes or clinical evidences. Next. The first example that we see here is the BRAF V600E variant. This is a very well-known hotspot variant in NSCLC. The knowledge base leverages clinical evidence, that is therapy recommendations curated from clinical practice guidelines such as NCCN, ASCO, FDA, and EMA, as well as clinical information from PubMed literature. On the left-hand side, we can see a couple of therapy recommendations that have been populated from NCCN and FDA, which are called rules, which was described previously by Siddhi in some of the slides. Additionally, the details for each rule can be found on the right-hand side, along with an auto-synthesized interpretation. The classification assigned to this variant by default is tier 1A, as the clinical data is populated for the patient's disease, which is NSCLC, from the expertly curated guidelines. This indicates this variant is of high clinical significance. The slide overall represents an extensive list of available therapies that can be considered for this patient due to the presence of BRAF mutation. Next. Here, we have a novel and rare structural alteration in the RET gene, which is the NPPA RET fusion. Although this alteration may not have been reported in the literature, but as Siddhi explained in previous slide, the knowledge base has the flexibility to curate clinical evidence or for RET gene only, with the clinical evidence is curated for the RET fusion occurring in NSCLC. As a result, this alteration is also classified at tier 1A, similar to or like BRAF. Next. Along with the therapy options, the knowledge base also provides insights into prognostic significance associated with the variant or alteration and the specific disease in question. For example, here on the left hand side, we can see prognostic information which is populated from PubMed literature. A point to note here is, in this slide, we can see that this alteration is classified at tier 2C. This is because the clinical evidence populated here is from PubMed literature. However, in the previous slide, we saw that the variant or the alteration was classified at tier 1A based on evidence which was populated from expertly curated guidelines. As per our default system, the higher classification trumps over the other classification. Hence, this variant will be classified at tier 1A. Next, we have a variant in NRAS, which is Q61L. Now, looking at this slide, we can see that this variant does not have any clinical significance or clinical evidence which is populated for NSCLC. However, there are some therapy recommendations in expert guidelines or expertly curated guidelines for this variant in other somatic cancers like cutaneous melanoma, colorectal cancer, and so on. As the clinical evidence available in the guidelines is for other tumor type. So as per the classification system which was explained by Siddhi in previous slides, this variant will be classified at tier 2C because the evidence is in the other tumor type and not in the patient's tumor type. Next, hence this variant becomes a variant of potential clinical significance. Now here we have the tier 3 and tier 4 variants. First is the ABL variant, which is in the highlighted section. It is classified at tier 3 as there is no clinical information available in the knowledge base for any of the cancer type. However, the minor allele frequency for this variant is not greater than 1%. Hence, this variant will be considered as a variant of unknown significance. Therefore, it will be classified at tier 3 by default. And the last two variants, they are classified at tier 4 or at the lowest level due to the high minor allele frequencies as observed in the nomad total frequency population column. So if the minor allele frequency is greater than 1%, therefore these variants are considered as polymorphisms and hence they are classified at tier 4, which are the benign variants. Next. Here we see a snapshot for this entire case, wherein a clinical summary gives a brief idea about the therapy recommendations available for this patient. The lower half of this report gives a glimpse of all the variants ranged in ascending order of the tiers, their clinical impacts, and if there are any clinical trials for which the patient is eligible based on the genetic alterations and the disease match. The point to note here is this is just a snapshot of the entire report. Therefore, only a few details are visible here. Next. Now, in the previous case study, we saw how knowledge base can help to identify clinical outcomes associated with independent variants or alterations. However, often we see that the clinical outcomes associated with independent variants or alterations are different when these alterations occur together with an additional variant or alteration, either in the same gene or some other gene. In the next few slides, we will see how knowledge base can help the users to assign correct clinical impact, especially for such co-occurring variant scenarios. Here we have an example of a patient who has NSCLC and who has been detected with EGFR L858R variant. Now, some of you might know that this variant is a sensitizing mutation. Therefore, as per the evidence which is available in the knowledge base, this patient is expected to respond to EGFR inhibitors like afatinib. Next. However, for example, this case, for a patient, NSCLC patient with EGFR L858R and an additional secondary mutation in EGFR, that is T790M, as per the evidence populated from guidelines, this patient is expected to be non-responsive towards afatinib. This, again, some of you might know that the secondary mutation T790M makes the patient or makes the cells resistant towards the first generation EGFR inhibitors. Next. Now one may ask, okay, this patient is not responsive to first generation therapy. So what can be the available therapy options for such patients? So once again, from the therapy data that is leveraged from Knowledgebase, from expertly curated guidelines, we see that these patients are expected to respond to osimertinib. Osimertinib is a third generation EGFR inhibitor. So these case studies exemplify how our knowledge base enables clinicians to take informed decisions considering both established biomarkers as well as novel variants, which ultimately enhances the patient care and management selection. Next. Now, as per our user feedback, our users find immense value in using the shared interpretation as a foundation when approaching unfamiliar biomarkers. These shared interpretations serve as a helping starting point, enabling them to draft their own interpretations with greater confidence. Additionally, our users rely on these shared interpretations to validate their own conclusions by comparing them with the interpretations and classifications decisions made by their peers. This collaborative approach serves as an effective gut check mechanism, ensuring the accuracy and consistency of their biomarker interpretations. Next. So now we come to an end to the webinar. Before taking the questions, let's have a quick recap. We discussed how the knowledge base is structured with all of its components, as well as what are the quality measures which are used at Velsera to maintain the content accuracy and comprehensiveness. We also saw how the KB or the knowledge base provides healthcare professionals with comprehensive clinical data access, intelligent decision support, and collaborative knowledge integration, which enables them to make informed decisions, optimize the patient outcomes, and enhance the accuracy of the reporting.